1 meter, 1 meter and a half. Like me, more. Imagine something like me that need to pack in a structure that is in the order of micrometer. Micrometer means 10 to the minus -6. So one thing, 1 meter and a half, packed inside a structure of 10 to the -6. This implies what? That we need to pack the DNA around protein that we call histone protein. DNA plus histone protein form the chromatin, and the chromatin at the end we pack it to form the chromosome. How many chromosomes do we have in the cell? 23 pairs. In total are 40. Muy bien. Seguimos.
Within the DNA are sections called genes. These genes contain the instructions for making proteins.
So, if I have to define a gene, what is a gene? A sequence of okay, DNA that codifies for a specific protein. Muy bien. Where we find gene? In the chromosome. And, okay, this we will study more in detail in biology, but I prefer that you know this concept. Chromosome contains genes. And the precise location of a gene is called locus. Locus is one, loci is the plural. A word that derive from Latin. Good. I know, I always do the same. That's why I hate. Okay, aquí estamos, ¿no?
3 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene.
RNA polymerase. Mierda. RNA polymerase because we have another polymerase. How is the other polymerase? Is the DNA polymerase. We know the difference between DNA polymerase and RNA polymerase. The DNA polymerase carry out the replication, also called
duplication
of the DNA. The RNA polymerase make the transcription producing the mRNA. Then now is like this. Aquí. Aquí.
RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus.
Look.
The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription.
Now look how move the mRNA.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
How we call this process in which we remove sections? Have you studied this? Say? Say. Splicing, okay? This mRNA, imagine, is a raw material once it's transcribed. We need to eliminate region of the DNA that do not codify for protein. How we call this region of DNA that do not codify?
Introns.
No. Introns. The exon, yes, codify. So imagine, this is my mRNA, I have an exon, an intron, an exon, an intron. The splicing remove the introns because they do not codify for anything. This video is incomplete because in the maturation of the DNA we have also the capping at the 5' ends and the poly-A adenylation at 3' ends. But don't worry about this because it's not part of the biochemistry process. Okay, now let's see how this mRNA...
The messenger RNA then moves out of the nucleus into the cytoplasm.
Okay, you see this? Doesn't matter. The RNA travel from the nucleus to the and move through the, how we call this structure? The nuclear
Pores.
Yes, nuclear pores. Okay. Da, da, da.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed.
Splicing.
This involves removing and adding sections of RNA.
We remove introns.
The
Now,
messenger RNA then moves out of the nucleus into the cytoplasm.
now look.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20...
You know this? Okay, for the synthesis of the protein, we need the mRNA that have been produced in the nucleus by the process of transcription. Then it's travel to the cytosol where find out the
Ribosome.
ribosome. You know that the ribosome scan the mRNA until it find out what? The start primero. First start and finally the stop. The start codon. When the start codon, how is it? The start codon is the pre-codon. What is, you know, AUG. AUG is the start codon. Small subunits of the ribosome scan, it's like this. My hands is the small subunit. Scan the mRNA until find out the AUG. Now the large subunits join the small subunits. And once the large subunit join the small subunit, the first tRNA that carry what? The amino acid, because we are talking about the synthesis of the protein. So this tRNA will bind the codon, will bind the mRNA. A second tRNA will come, formation of peptide bond, and so on. Okay, look at the video.
20 different types of amino acid
Sorry, here we have, oh, a moment.
to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids.
Look, this is the
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, now go to YouTube, guys, annotate the video, and then based on your notes, because this will help you to understand these processes in your mind. When I studied, there will not this kind of video that we have right now, and for me it was very difficult to imagine the synthesis of the protein, but this video, you know, is very clear. It is 2 minutes, so it's fine. I hate YouTube because people to explain one stupid thing like 30 minutes. I don't know if you have looked at this video that talk about Excel or something like this. 30 minutes to explain one thing. Okay, so the ADN a protein, young genome. Okay, fine. And now we go back. So why we say all this stuff? Because in your mind you need to have clear that we arrive to the structure of the protein with this process. Now, do we know in which part of the cell the synthesis of the protein takes place? In the cytosol. And which are the main actors of this process? mRNA, tRNA, ribosome and amino, and these amino acids are attached on the tRNA. No tRNA, no synthesis of the protein. Yes? Okay, now, if we want to write the structure of the amino acid, let's make this exercise together. Okay, what I have to write? Structure of amino acid. How is
Carbon,
carbon,
hydrogen,
hydrogen,
carboxylic group,
carboxylic,
amine group, and R group.
and
R group.
what is the meaning of R group? Is the lateral, and this lateral chain, what is the meaning of this lateral chain? It is a functional group that is different or is specific for each. Okay. How many amino acids do we have in nature? 20. So how many tRNA we will have? 20 as well. Okay, this is the form with both NH2COH. But what I said in aqueous solution, how is this amino acid? In which form we find? In the sweeter ionic form. How we write the sweeter ionic form?
COO minus.
The NH3 is
Plus.
CHRCO minus. Okay, so at physiological solution we find the amino acid in the sweeter ionic form. Actually, we will say that this form doesn't exist in the real life. Okay, so now we start last time, no, to talk about. So depending, okay, considering that the amino acid have the amino group and the carboxylic group, they will be always polar molecules, okay? Because we know that the carboxylic and the amino group are polar because they can form hydrogen bonds with water. Okay, so now depending of the lateral chain of the amino acid, depending of the lateral chain, we can find lateral chain that are non-polar, lateral chain that are polar, lateral chain that are aromatic, lateral chain that are positively or negatively charged. This makes the amino acid hydrophobic. No, guys, all the amino acids are soluble. Estamos? But then this solubility will change on the base of the lateral chain. Of course, a methionine will be more insoluble than the tyrosine, for example. Now, do we need to memorize all these lateral chain? The answer is no. But you need to have the instrument, the tools to say this amino acid has a polar lateral chain, and polar lateral chain. Let's analyze one by one. Okay, so if an amino acid has a non-polar lateral chain, which functional group do you expect that this amino acid will have if there is a non-polar? The non-polar, excellent non-polar functional group is the methyl group. We can have one methyl group, two methyl group, or an association of them. In fact, here in this table you have all of them. But now let's look. We have glycine, guys, this one remember because it's the only one that has a lateral chain, the hydrogen. Then we say non-polar, methionine, alanine, it has a methyl group. The proline has a many methyl group, for example. Then we have valine, 3 methyl group. So when you see only methyl group, do not attach it, for example, with the amino group, just methyl group, you will be able to say this amino acid has a non-polar lateral chain, as in all these cases that you can see. Okay? Now, aromatic is easy to remember. Aromatic means that they have a cyclic structure, cyclic form. How many aromatic do we have? 3. tryptophan, tyrosine, and phenylalanine. Okay, what I say always, when we have a cyclic form like benzene, the molecule is non-polar. But here we have an exception. What is the exception? Is the OH, how we call OH, hydroxylic group, present in the tyrosine. So, tyrosine belongs to the aromatic, and it is the only one that is polar because of the hydroxylic group. These amino acids, aromatic like tryptophan, tyrosine, they have a property. Okay, they can absorb the ultraviolet light. Okay, it is a property. Where we use in a laboratory this ultraviolet light in an instrument that is called a spectrophotometer. Do you know how visualize the spectrophotometer? I will show you. Okay, why we use the spectrophotometer? Do you know? See or no? No?
Quantify protein.
To?
Quantify protein.
We can quantify protein. We also can quantify, for example, the number of cells that we have in a culture media. Okay? Spectrophotometer. Okay. Imagenes. Okay, guys, this is a spectrophotometer. This is this instrument, but I want to show you. You know what is a cuvette? Yes? No?
Yeah.
Todo el mundo? Okay, guys. Imagine I am in the lab, no? I'm working on a protein. Do you know there are genetic techniques in which you can take the sequence of your protein? You can overexpress. Overexpress means you make a lot of copy, and then you can purify this protein to have a pure protein. But why we need to have a pure protein? Because if in a lab you are studying the specific function of a protein, you need to have pure protein that you will combine with specific substrate to analyze this function. Okay, stop. So we know the sequence of our protein because otherwise we cannot work in the lab with the protein. We extract this protein, we purify the protein. Now we want to know its concentration. Why I need the concentration? Again, to make the calculation for the in vitro experiments. Okay, in vitro, you understand the meaning of in vivo, in vitro. In vivo means what happens is in the cell, in vivo. In vitro is under laboratory condition. Okay, so I put my protein in this cuvette, and then I put the cuvette in the spectrophotometer. I set up the wavelength at 280 only if I know that my protein contains aromatic amino acid. If in the structure of my protein that I know there are not aromatic amino acid, the spectrophotometer will be used to quantify the protein. The answer is no, we will use another methods. Okay, I explain all this stuff that you feel consider very boring to explain the practical application of this property of these amino acids. We know this. They are able to absorb this ultraviolet light. So, this property helps us to take advantage from this property to measure the concentration of our protein. The spectrophotometer, guys, is a very easy instrument to quantify. The other technique like Bradford method there are, or formation of a standard curve is more boring. In this way, put and we measure the concentration. Okay, so polar amino acid. Which are the functional group that make this amino acid polar? Which are? Hydroxylic. Yes. Then we have the sulfhydrylic group present exclusively in the cysteine. Then here what we have? How we call this how we call amide group? Okay, all these groups are polar. But look, here you will say, but we have a methyl group. Yes, but when the methyl group is with amino, carboxylic, oxidrylic, this lateral chain will be polar. Okay? If we have only methyl, like in the case of nonpolar, they will be nonpolar. But if we have methyl group in combination with sulfhydrylic, amide group, hydroxylic group, this amino acid will have a lateral chain polar. ¿Estamos? Vamos. I don't want that you memorize any amino acid. Only glycine, that is the easiest one, and tyrosine, okay? The rest, by looking at the structure and the functional group, you will be able to say is polar, non-polar. Now, polar and these are uncharged. We have here formation of disulfide bridge between two residues of cysteine. Remember that this chemical bond is how? Weak or strong? Is strong. It is a covalent bond. Guys, they are the disulfide bridge, the ones that are break or formed when we have different type of hair. Now I don't remember if in curly hair we have if it's reduced or oxidated. Now I don't remember, but it's the oxidation of the reduction of the disulfide bridge that determines the type of hair that we have. Okay? Positively charged. What we know about the positively charged? Look, in this case we have a methyl chain, but we have the amino group. Amino, amino, and the structure with amino. Moreover, they are positively charged because they have a positive charge. So, if we have amino group plus the positive charge, this molecule, how is? Totally polar, okay? Positively charged with amino group. Then we have negatively charged, very easy to remember. There are just two, aspartate and glutamate, also called aspartic acid and glutamic acid. If we say aspartic and glutamic acid, is because which functional group they have? The carboxylic, amigos. Aspartate because it's the ion. If it's aspartic acid, we will have COH. Considering there is the negative ion, will be aspartate. Negative charge, carboxylic group, totally polar. So remember, like this. We have polar amino acid, there are the one, polar, but polar are also white. Which ones? The positively and negatively charged. All of them are polar. Then we have the non-polar, and then we have the aromatic. Within the aromatic, two are non-polar, and one is, which one is the tyrosine because of the OH. Ya está. Okay, I go very fast with this group. At the end, this will be my favorite. We don't know yet. I always have a favorite group each year. Last year was the M14. They were very good, very good. They were also very kind. Were really very focused, and also the grades were brilliant. 9, 10, maravillosos. And the M14, I stayed with you the entire year. They were okay. But it was not my favorite. You know, this year I am still deciding which one. is my favorite group. Okay? I don't know. M11, not for sure. M11 is always a bad group, not because they are bad people, because they have a terrible timetable. They have always lesson at 12 until 4 or from 10 until 4:30, so they are always hungry. You know, they are not focused because they don't eat. I mean, for me it's important that you have time to have a lunch or a brunch before going to the class. Then people arrive always late, like someone here in this class today, okay? They always arrive late, always. So, of course, imagine myself with the fire, okay? I always arrive 5 minutes late. Why? Because it gives you the opportunity to arrive more or less. But then, okay, they were very terrible. And the M13 was, a good group. Do you know some of the girls? Maybe do you know some of the girls from last year? No. But then also, I had a very good student in the M13. I met a girl, then she changed totally. Now she studied economics. Yes, from dentist. Yes, guys, because sometimes it's because, not because you choose to study dentist, it's because your father chose it for you, you know? And it was the case of this girl. But I saw her not interested in this subject, you know? And now, finally, I am really happy because she finds, her path, her destiny to study something that really likes you. If you study, the first 2 years in all these medicine, biology, dentistry, biochemistry are a little bit hard. But if you pass the first 2 years, you will enjoy if you like dentistry, of course, this is the base, no? You really enjoy because you say, okay, I finished all the anatomy, physiology, biochemistry, biology, all the big subject, and you really will enjoy dentistry. But in order to arrive at this point, we need to study this because these are the basic subjects that will help you to understand. For example, last week I met a girl that now is in the 4th year and was studying the type of anesthetic that you will use in the dental clinic. And there were anesthetic lipophilic, hydrophilic. So if you don't have this base of biochemistry, you will not understand a lot of stuff in the future. So my personal recommendation is just focus very hard these 2 years, study as much as you can, and then you will relax a little bit and you will enjoy. But life is hard, my friends, really hard. I work the entire weekend, 5 hours on Saturday and maybe 4 hours yesterday. Yes, because I'm also evaluator of postdoc program Marie Curie from the European Commission. Marie Curie, you know what is? There are fellowship that you can ask in the European community to make PhD and postdoc. So I am evaluating the project for students that want to make this Marie Curie. So I had like six proposals that I have to study and to say it's good, not good, why, okay? So during the week I don't have time to do this because of the class, because of the research and so on. So I just close myself 5 hours alone on Saturday. This is because I really like my work. Because if imagine if you don't really like and you have to do, of course they pay me, it's not for free. Yes, but doesn't matter. If you don't like, it will be very hard. Okay, so you need to follow. I will not say you follow your dreams because sometimes we don't know which is our dream in life. For example, I imagine myself teaching. I was making PhD, I really like laboratory research, and I still love this part. But I had no idea that maybe I really like this teaching. But I never thought about teaching. But now I will say, okay, I really like, okay, as you can see, the way in which I transmit my passion for biology, biochemistry, because it's not easy to explain this kind of subject and create an engagement, which is not easy, you know, it's not easy. But I think that is extremely important that you have really few concepts that you can save and apply then in the other subject of the dentistry. So, just closing these parentheses, we move to the amino acids. They have optical properties, okay? They are electric dipoles and amphoters. What is in this imaginary glossary note that we are making? Amphoteric compound means what? They can act as acid or base. In this imaginary glossary, amphoteric compound. In this imaginary glossary, which example of amphoteric compound we made? Water and now amino acid. If I ask you, the amino acid are soluble molecule, what you will reply? Yes, but the solubility can change depending on the lateral chain. Guys, there is like hay un poco de tierra. Okay, optical property. Do you remember this carbon alpha? What is the carbon alpha? Is the central carbon. Okay, why we say we have L and D amino acids? How we call these two molecules when they are mirror imaging? How we call this molecule? Enantiomers. We have enantiomer L, enantiomer D. And this L and D depend on what? On the position of which group? The amino group. If this amino group is on the left, we will have L, alanine, and in the case of on the right will be the alanine. Okay? Amino acid exists in two forms. Okay. Chiral carbon. This is another concept that we will annotate in our imaginary glossary. Okay? This chiral carbon. What is the in chemistry? What is the meaning of chiral? Is a molecule that is bound to 4 different substituents. Okay? In the case of glycine, the carbon is chiral or not?
No.
No, why not? Because the two substituents H and H are the same. So we can state all the amino acids are chiral except the glycine. Glycine that belongs to the non-polar. Muy bien. Now we analyze the amino acid as electric dipoles. Okay, so this is what I wrote before. Non-ionic form, we have an H2CO. You understand? COH and H2. The sweeter ionic will be an H3 plus CO. Okay, yes? Now, we start with this one, okay? We say that the amino acid, like the water, nothing. So October is the worst month of the year for me because I have to pay a lot of stuff for the car. The insurance, you have to pass the inspection. Do you know how to say?
The maintenance.
I don't know how to say in English. Imagine, if you have a new car, you have to pass this inspection after 4 years. After 4 years, you have to pass this inspection after 2 years. And after 6 years, you have to pass this every year. So 200 euros to say your car is okay. You know? ITV aquí se llama.
Inspection technical.
Inspection. Yes. Now they send me 200 euro. Okay. So this is, look at this graph. What we have? We have the pH and the concentration of what? Of the amino. Okay, so if we don't know how to read this graph, I help you, I guide you. We start from the, which is, I say, the form in which we found the amino acid in solution, the sweeter ionic form. Are we able to distinguish which is the sweeter ionic form between these three? Is the one in the, because of the NH3 plus and CO-. This is our starting point. Then, here we have what happened from this form to that form. This implies what? What we have, what is the difference? We have the CO minus will be proton. So assume, so when a molecule capture a proton, behave like a base. And on the contrary, what is the difference between the blue and the green? It's give up a proton. Which proton? The one present in the amino group. So when you lose a proton, you behave like a... Okay, so now let's see when an amino acid behaves like an acid or like a base. In general, it's easy to remember. Okay, in acidic solution, what is the most prevalent form? The one there is protonated. So means at lower pH until pH, until pH 1 and 1/2, what is the most predominant form? It is the one that is protonated. Okay? So in this case, in acidic solution, the amino acid behaves like a base because it captures the protons. Okay, until which point we have this one, and the sweeter ionic starts to increase its concentration from 2 until we find a maximum between 4 and 8. Okay, between 4 and 8, the most predominant form is the sweeter ionic form. When we move from the 8 until 12, 14, means what? That we are moving to a alkaline solution. Alkaline means there is a basic. So if the environment is basic, the amino acid behave like a acid by releasing the proton. So it's easy to remember. I am in neutral condition between, or better say, between 4 and 8. The most predominant form is the sweeter ionic form. In acidic condition, the amino acid behaves like a base capturing the proton. In which part of the amino acid will capture the proton? In the carboxylic group. When the pH became basic, the amino acid behaved like an acid, losing the proton present in the amino group. Clear? Okay. Here you have another slide that say the same. Look at this. Again, if we don't know where we have to start, we start always by looking at the sweeter ionic form. In this case, which is the sweeter ionic form? The one on the left or the one on the right? On the left, because we have CO minus and H3. In this case, the pH decreases. Means what? That we have a acidic pH. The pH decreases, so in the pool of this solution we have a lot of H+. So if we have abundance of H+, the amino acid will capture these protons, okay? Behaving like a base. And here is the opposite. pH increases means that we have more OH minus. This OH minus will capture the protons of the amino group. Yes? When you have a basic environment, imagine that you have NaOH dissolved. So NaOH, we know that dissolved in Na plus and OH minus. So you will have in this solution a pool of OH minus that want to capture the proton. Okay? Capture the proton and we have NH2CO minus. Okay? Clear? Is easy, no? Is it easy if you understand? If you don't understand, then you will see this slide and you will say it is more difficult than Chino Mandarino. Yes? Do you speak Mandarino?
Yeah.
This is your... mira, ¿ves? Because it's the most spoken, no? In China.
Yeah.
Yeah, okay. Yes? El habla chino. If you want to learn Chino Mandarino, our friend can help us. Yes? How you say hello?
你好.
Ni hao? Yes. Está bien. ¿Qué pasa? Okay, so this is again the same, but now we focus on the charge. When many hours sit, you know. Today will be yoga day. I need. My body is saying, okay, you need some stretching. Okay, now regarding the amino acids. So when a molecule captures a protons, how will be the net charge? When you capture something, you will be positive, +1 if you capture 1 proton, +2, 2 proton, and so on. When you release or when you lose, you will have a negative charge. Okay, again, our starting point, if you look at this slide, what will be? When you look at this slide, our starting point will be the sweeter ionic form or physiological form. So we say that in acidic environment, the amino acid work as a base, capturing a proton. So if the amino acid capture a protons, the net charge will be, when you capture, so you have something more, the will be +1, positive. And in the case of, what happened with your hair today? So people just wake up and come here. Yes, you wash your hands, your face. Guys, what about you? No pajama, no? So imagine, no? You arrive to the dental clinic and the dentist is with this hair, with the pajama. What do? You will say, okay, baby, goodbye. Yes? Guys, when we have working with the public, we have to take care of our aspects. Yes? Do you think that we are stupid? Wake up early in the morning, shower, make up, and everything, the shoes of the clothes, so it's not casual, you know? I have to think every day where I have to put. But then there are people that just wake up and change. Muy bien. Ok, yes guys. Ora, vamos a cortar un poquito. Ok, no pijama, please. No pijama. But you are always asleep, so I mean, are you sleeping well? No, not very well. Se nota, I can see. I can see. So, important, so I mean, I don't mind if he came with a pajama, but we are at the university. We cannot go to the class in pajama, guys. Yes, I mean, you have to take care of your aspects because in the future you are going to work with clients, with patients. Okay, so this part is also important. Okay, so we say amino acid that capture a proton plus 1. Amino acid that releases a proton minus 1. Okay, and now we move to the pKa. What was the pKa? Do you remember how we can define the pKa? pKa is the constant, the minus log of the dissociation constant of a weak acid or a weak base. The amino acid, how many pKa they have? Look at this table. How many pKa the amino acid have? Huh? All of them, they have at least 2. And some of them have no 1. 3. One more.
One more.
Yes, one more. But it's depend on what? The pK1 refers to the dissociation of the read COH, the pK2 refers to the dissociation of the, and the pKr to the lateral chain. Now, which are the amino acids that have a lateral chain with the pKr? It's depend on what? On the fact that they can release or capture a proton. And which are the functional group that can release or capture a proton?
Aromatic.
OH that is present in tyrosine. Then which are the functional group that can capture or can release a proton? Amino, carboxylic, and OH group. Okay, if we don't know, let's look. Tyrosine, which is the functional group of the tyrosine, is the cyclic form plus the OH. The OH can release a proton, yes. Now we move to cysteine. How is the cysteine? The functional group is, okay, go back to the slide. How is the functional group of cysteine? Let me check. CH2, no? SH, which is the lateral group CH2, SH, which is the one that can release the proton, the SH. Then we have the lysine. What is the functional group of the lysine? Is the, we have 4 methyl group and NH2. So again, NH amino group. So if I ask you, what is the condition by which an amino acid has a pKr? He needs a functional group that can be ionized, means that can lose or can capture a proton. The methyl group can capture or release a proton? Totally false, okay? So the non-polar amino acid, they have the pKr? Never. You can double check, okay? Methyl group cannot be ionized. CH3. cannot be ionized, cannot release, neither capture a proton. And now we go to the titration curve. Do you want to stop? 5 minutes? Okay, we stop. Okay, I have a session when I see with men's hair. When I see they are too long, I always say you have to cut. Yes. That is not for you. So it's in general, it's an obsession. You know my brothers maybe go once a week. That's a lot.
Where were you yesterday? Where were you yesterday? I was when I saw him, I recognized him. He looked like Yo, Sean, I heard there was a fight. Huh? You got into a fight?
No.
Someone told me you got into a fight. Oh, you should have come yesterday. We went to Burger King. That guy's house. You and yeah. And the other guy, the Albanian guy. Yeah, Ray.
He told you that it was like you're not
yeah, I didn't know it was like I knew who it was, but I didn't know they better know. I'll come and
autonomic physiological stress responses among the fish, a heart rate variability and psychological assessment study say that many dentistry teachers so how male and female respond or cope with stress. I like you about Can I go to the toilet? Yes.
This was like girls.
No, I have to go to the toilet.
You're sick? No, I'm just like too tired.
Oh yeah. Today we're gonna see. Wait. Oh my God, I've got to come to my bag. Hold on.
I was wondering, like, this Wednesday, like, when she, like, sees the assignment thing, the proposal draft,
Yeah.
I think she's going to tell us all. No, like, my group.
Why?
Because, like, she said only the group leader can send it, right?
You sent it.
No, like, you should send it, but then, like, 3 people on her group sent it. 3 out of 4 people.
Why?
I don't know.
If the group leader sends something, it's fine.
Yeah. And then I think I raised submitted it, so I did it twice.
No, she won't be. No, I don't think she'll be.
What are you going to do for like? I know this is very childish, but what are you going to do for Halloween?
Nothing.
I know, nothing.
Nothing.
I know.
You?
No, nothing.
Right. You like, okay. I always dress like a pumpkin. Pumpkin. Like you cut the pumpkin head.
Yeah.
A hat. Out of a real pumpkin. It's
Yeah, that's nice.
but we used to like carve pumpkins.
Mais il était là, espagnol, j'étais loin de l'espagnol.
C'était un massage pour le chevelu, frère.
So like,
On n'a pas fait les
Oh là là.
Il commence à m'envoyer parce que c'est un mec qui est marié, qui est bordelot et non mais il était pas là. C'est quoi ta question? By the end of the day, non mais tu peux I think of this man. I like the accent. Elle est où? Lui, ça va, il est chez moi. Mais oui, en vrai, il est près de mon frère. Oh, bah c'est elle aussi. Non, il gagne sa réponse. Et j'étais défoncée, il m'a dit qu'il m'avait vu à la chose.
so like, when I was in primary school, like there was this guy, and then he like cut a pumpkin, like that pumpkin head thing, but then he put like speaker into it, and then it's just like it just like played everyone's name at random intervals. And then he put a light bulb in it as well, so like when he speaks, it suddenly lights up.
light bulb.
Yeah. So when he speaks, it lights up.
You know when you carve pumpkins and you put like a candle in it?
Yeah.
It stings.
Yeah.
What the fuck? Then I feel like you're supposed to try them first.
What?
I used to carve the pumpkins, and then I put like a candle in it, and it stings.
Yeah.
You're supposed to like let them dry out,
Yeah.
so that it doesn't sting.
No, I didn't.
So yeah.
Français, anglais, espagnol,
Do you speak French?
Yes, I do speak French.
Stop making pictures. Stop publishing stupid stuff. Mira, tú que hablas español. No. Yesterday, at the Spanish television, there was a documentary about the like teenagers that have a strong addiction to the mobile phone. Mira, ¿tienes tipo? ¿Sí ves? No, pero nunca. Bueno, da igual. Es un documentario de la Sexta. La Sexta del Canal 6. Ay, cuidado. Okay, so there was. Do you remember last year that was the trials against Facebook? Okay, in this interview, in this documentary that I watched yesterday, there was like the chief of the security of Facebook. They said that at the beginning, when Facebook born, like 2009, the security was ensured for for people. But now, also, if you want to report, for example, video violence or pederast and so on, maybe you say report this video, but actually, this engineer said that this option is totally fake. That you think that you are reporting something but it's not true. And yeah. What he said yesterday. And then there was like they talk about with teenagers with strong problems. These teenagers pass like 18 hours, okay, on TikTok, on Instagram, or just playing with their PlayStation and so on. 16 hours. Okay, do you know what is the number of hours that make you an addicted?
No. Yeah. 8.
Okay, there will be more than 30 hours per week. So, no, per week.
Yes.
How is your average per day? 7. So you are addicted because 7.
All the lessons are on our phone.
Just for the blue light at night.
Non, mais du coup, ça veut pas quand tu révises.
How can I see the, you know how is the bath?
Yeah, over there. Just have to swipe to the left.
Okay, now we put in common.
China? Or you're from Spain?
From China.
From China, and then you moved to Madrid to study or
Study.
London studies.
Yeah.
And at this university or before you?
Before.
Before, okay. Because the job says that she's from France, but both of their parents are Chinese and make in the parents. Elle regarde ton écran, alors je dis pas.
Bueno, education.
And also I call my boyfriend
Because now
Alors, how many?
No, I'm scared. I know someone like is always online.
Yeah.
I texted him in the morning at 7. Then I spent my day, I slept. I woke up at 4. This bitch, he was still awake at 4 in the morning. Yeah.
Madre, I want to see mine. Yesterday. Bueno, 4 horas.
Yesterday.
Why does mine
So, 5 hours on Snapchat. ¿Qué se hace en Snapchat? I don't have So, with an average of 8 hours per day, multiplied by 7, but you don't make any videos. And you?
I was. My mother will kill me. She. Mama,
Your name? Please, this is very serious. I want to have this conversation with you.
That's why he gets dressed in the morning and he comes with his PJ.
Monday 30 de septiembre, que fue el Monday de la semana pasada. He stayed 3 hours on TikTok, 2 hours and a half on YouTube, and 2 hours and 15 on Snapchat. Instagram only 45 minutes. Pero tú te das cuenta, mi amor, que así no se puede seguir. We cannot. Traduce.
Moi j'ai bloqué mon.
Today, guys, it is 12. 6 hours.
Last night.
Ya. He must sleep.
Moi j'ai bloqué mon thème.
I see your face, and I can say, this guy is not sleeping well. Okay, so. We are talking about hours that we spend on the mobile phone. Our friend, we don't mention who is from this morning, it is 2 o'clock, just 6 hours and a half in the first part of the day. Okay. Because yesterday I was watching a documentary, okay, regarding teenagers and the addiction and the addiction to the mobile phone.
How much?
Okay, do you know what they do? Imagine young people that you
No. Like 6 hours.
6 hours this morning.
No. Back in general, innit?
Then 6 hours.
That's stuff I least 10 heures.
Yes. So, okay. Do you know how we solve this? No,
Yeah. You can do applications.
You know how we solve this?
Having many activities.
First step will be eliminating which application do you think that take
Instagram.
TikTok?
Yeah.
Guys, I use TikTok, but you know where? No, Vera. No.
TikToker.
No.
Yeah.
Do you know where I use TikTok? For example, I am, for example, in Paris, and I look for a restaurant, nice restaurant, done. Or I am in, like to have information, or recipe with, I don't know, pasta, whatever. Or best restaurant in Madrid. Only in this case. Only in this case. Vera. People with my age, we don't use amigo. People of my age, we don't have Snapchat, so problem solved. You know? This Snapchat, I see that use a lot of your energy. What do you do? 3 hours, 4 hours on Snapchat. Estamos locos. So, TikTok, fuera. Snapchat, no, because I think that you communicated, with Snapchat. Okay, so in this documentary, actually, teenagers with this addiction, you know what they do? They went to a place with psychologists, educators, and so on. They remove the telephone, no tobacco, no telephone. They do everything at home. They just wake up. They have a shower. They clean house. And then they work gym three times per day. Gym.
Per day? 3 days.
Once a day.
That's too much.
1 day per week? No, 1 day. So you must go to the gym every day.
No.
1 hour a day.
You are too busy.
Go there and run. You will see, no problem. Guys, we cannot stay home by looking at the mobile phone like amoebas, you know? Okay, I will explain something that is physiological. Okay, imagine that right now I publish a story on Instagram, no? And then I have no notification. I don't want to see. Who is your dad?
Yeah.
Okay. The notification, I remove the notification. This means that you put like on my story, I don't see, unless I go inside the program. This in our body, in our brain, generates what? A circuit that is similar to the addiction of drugs. Okay? So, when you see that people put likes, how do you feel? How do you feel? When you post something and you receive a lot of likes, how do you feel? Be honest.
It depends on who likes.
Okay, but in general, you feel happy. But you know why? Because in your brain is generating. You know what? I will not see. Will generate what? Dopamine. And this is the same that our brain releases when we take like cocaine, heroin, is exactly the same. Okay? So this like that this girl put on your post or your story will produce the same pleasure the cocaine or heroin or whatever produce in our body, in our mind. Also, you are growing up, no? Taka, taka, taka. Your brain is full of neurons. Pay attention, this is more important than a biochemistry class, I think. Your brain has a lot of neurons, but having number of neurons does not mean anything. But what we need to create? Synapses. And how we create synapses? By studying, by learning new things, by speaking a new language, by learning the language, for example, of the music, by making something with our hands, como se dice, manualidades, okay? For example, in my case, I always have in my mind, I want to dance, but I see that I have no coordination. Why? Because I used to do other kind of activities, but not dancing. So when I dance, I say, oof, no estoy coordinada, I'm not coordinated. Why? Because my left and right part of my brain were not connected. Then, with practicing and practicing, of course, I don't see myself as a dancer, as an étoile, okay? But I see they can overcome, okay, these limits. But what do you do to overcome your limits? Watching Snapchat. And this will produce what? The death of many neurons and the impossibility to create new synapses. And if your neuron deads, this means what? That you can suffer from Alzheimer's disease. Hey, amigas! You can suffer from Alzheimer's disease. We understand this. So then when you say, I don't remember. No, my friends, you cannot say I don't remember with 20 years, with 18. Okay? So, what we can do, when we arrive at night, we start to avoid this light. The blue light is killing us. When I go bed, I do like this. I put the clock and I put like this my telephone. I never put on this side because I see the notification. So I do like this. When at night I wake up because I need to go to the toilet, I don't see the hour. I directly go to the toilet and I go back. Why? Because I don't want that this light will, wake up me or whatever. You can start to do like this. Go to the gym, go to run, or leave the mobile phone at home. I know that is complicated, but you have to do this. This is an exercise that will help us to live better, because we cannot stay 8 hours, okay, just looking at mobile phone. It's too much. 6 hours is too much, okay? Yes? Also listen podcast. I have this great passion, the podcast. So just listen, relax yourself, don't look. You know that you can also have problems with your view because of the screen. Yes? You have, no? Yes? So let's make this exercise. I want to make this exercise. In class, please, just put the mobile phone in your bag. Just focus on what we are doing. This will be a very great exercise for all of us, also for me included. Okay? Of course, if you are waiting for an important call, you can put the tone, and if someone is calling you, you say, Mariangela, it's important. Okay, fine, no problem. I remove the vibration when I receive, for example, WhatsApp, because this makes me nervous. Zoom, 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 zoom. I remove everything, and now I will say, I'm better. At night, I sleep. Finally. See? Corre, run, make sports. Do this. We cannot be like a zombie, you know. Okay, now, I explained the titration curve. Now I will give you back once we finish. We can stay 10 minutes without the mobile phone? Yes? You can give me your as well for these last 10 minutes? Muy bien. Perfecto. Next time I will take a bag, and in this bag we will put all the mobile phone. Yes? But don't install the program on the computer. Titration curve. What is the titration? We say there is an experiment that allows us to determine what the pKa of an acid, but in this case we will calculate the pKa of the amino acid. Okay, again, if this could be a question that you can have in the exam. Imagine that you have this graph. This is the titration curve of which amino acid. Are you able to identify which amino acid is? CHH is the easiest one is the glycine. Okay, this is the titration curve of the glycine. Because of the lateral chain there is an hydrogen, the glycine does not have a pKr. The glycine has just the pK1 that refer to the carboxylic, the pK2 that refer to the amino, okay? And we have the pi, also called isoelectric point. What is the meaning of isoelectric point? Is the point at which the amino acid has net charge equal to 0, okay? Okay, let's see. Okay, now if I ask you, which are the buffering regions of the glycine? How many buffering regions glycine has? We say 2. Which are the range of buffering region? The first one was between 1.34 and 3.34, and the second will be 8.60 and 10.60. Okay, so now the isoelectric point, when the net charge is equal to 0, the net charge of the glycine, when it's in the sweeter ionic form, the net charge is equal to 0. When the net charge change into specific region. The first one, in the acidic region, when the COH will be protonated. So when we are talking about this point, in this point the net charge will be +1. In this other buffering region, the net charge will be -1, because in this case the amino group loses a proton. We make another example, a little bit more complicated. The glutamate. Okay, glutamate or glutamic acid, which kind of amino acid is? Is polar, negatively charged. We are able to identify this. Okay, if we have to make a circle in the lateral chain, which is this lateral chain? These are the symptoms, okay? We cannot have the mobile phone, and because it's la síndrome de abstinencia. You have coffee. Okay. Muy bien. No, which is the lateral chain of this? Look at the slide and say which is the lateral chain. Is this one, no? Muy bien. In this case, how many pK we have? 3. Why 3? The first one refers to the no. The first one refers to the eso. The second refers to the lateral chain, which is the group that is ionized. Is the carboxylic group, and finally the amino group is the one in the pK2. Okay, now if I ask you, let's see how change the charge of this amino acid. Okay, if I don't know, I start from the sweeter ionic form. Which form is 1, 2, 3, or 4? The sweeter ionic means what? That the net charge is equal to 0. So is the second. Okay, so okay, if we move to acidic pH, it will be +1. Between pK1 and pKr, we have net charge is equal to 0. Between pKr and pK2 will be net charge -1, and from pK1 until the end of the pH will be -2. ¿Estamos? Now, if I ask you which is the pH, which is the net charge of the glutamic acid at pH 1. Okay, pH 1 means here, no? Okay, what will be the net charge? This is acidic pH, so it will be +1. Okay? Then if I ask you pH +1 here, pH 3, there is between pK1 and pKr, will be? No, is between pH 3. The net charge will be? Bueno, if you have to decide between +1 and 0, you will say? No, no se puede, 0.5. This will be, no, 0 is when you arrive here.
So 0 is from there to the top.
Will be +1. If I ask you a net charge at 7, will be between 1 minus 1, muy bien. And then if I say pH 13, will be -2. Okay?
Where's 0?
0 is between here.
It's this point.
This point.
It's just the point, okay.
This point. Okay? This point will be 0. Okay, of course, more close we are. Yes, but this is the explanation. Okay?
Okay, I got it.
You got it?
Yeah.
PK1 from this point, okay. Between PK1 and PKr, it is +1 until we arrive to 0. Then between PKr and PK2 in this region, we have -1. And from this point, we have -2. Okay, if I ask you pH 14, the net charge will be -2. If I say pH 3, this will be +1. Okay, maybe we have another example. Okay, last example, and okay, but we will retake also on Friday. Okay, this is the histidine. This is our starting point, no? 0 starting point. Okay, taka. pH 7. What will be the net charge of histidine? pH 7 will be? Why is not -1? Because -1 starting from the pK2. So if I say pH 10, it will be? If I say pH 7, it's between
0 and -1.
0. Between 0 and -1, but it will be 0. Now, between pKr and pK1 will be pH +1, and then from pK1 will be +2. So if I ask you pH 1, net charge will be +2. Okay, then pK1, pH 2 will be +1 until the next, that will be the pK6. Okay, pH 8, again, 0. Okay, all this region is 0. All this region is +1, all this region is plus 2, and from this will be minus 1. Okay guys, we stop because you have to go to the building A, and we will see next Friday.
Merci.
Studio.